Hello, my dear students of class 12. Hope all are doing fine and very busy with your pre board examinations. I am here with another practical based activity video, that is of mitosis. I've already made a previous video of mitosis, but here I am going to show you what you have to do on the day of examination if you get the question of making a slide of mitosis. So, this is one of the you can see preserved specimens of the onion root tip, which has been treated earlier with the fixative, and then it has been preserved in 70% alcohol. So you can use the root tip which will be provided by to you by the teacher, and you need a stain for staining the root tip, that is the acetocarmine stain, and you need one normal hydrochloric acid that you have to prepare. So how to prepare one normal hydrochloric acid? So you have to take 96.5 ml. I have taken 96.5 ml of distilled water, and along with that I have taken 3.5 ml of hydrochloric acid. We have mixed it in the beaker. So here I have taken two measuring cylinders, one the bigger one for the distilled water, the smaller one for the hydrochloric acid, and try to use a pipette or a dropper for pouring the hydrochloric acid. So I have taken pipette. So while using all the glasswares, be careful that everything should be clean, and I'm cleaning all these glasswares in the tap water, and then after drying or rinsing thoroughly, I will use this glasswares. So rinse properly all the glasswares that you are using, and then you make it dry, and then you use it. So this is the thing that you have to remember. And now it's the time to make the one normal hydrochloric acid. So this is the distilled water jar. I am pouring some distilled water at random in the beaker, and then from this beaker I will pour it in the measuring cylinder. Here you need a watch glass. The watch glass is for staining the root tip. In the dissection box, from the dissection box I need three important things. One is the needle. Needle is required for placing the cover slip over the slide. We need a forceps for plucking out the root tip from the bunch that I have already preserved in this bottle. So what I have done is I have tied all the root tips together with the help of a thread, and then I have preserved it in the alcohol, 70% alcohol. So you have to just take out one of the root that you need to prepare the slide from out of it, and you also need a camel's brush for transferring the root tip from the watch glass to the slide. So before doing the activity, make sure that all the reagents, all the glassware, everything should be near you, so that you can easily do the activity without any hindrance. But if you do not make a plan of what do you require for the activity, it will be very difficult for you to run here and there. So before making all the setup, so you jot down what are the requirements. So here I am trying to make the one normal hydrochloric acid. So you take 3.5 ml of hydrochloric acid and 96.5 ml of distilled water, and pour them both in the beaker and mix it well, and that is the one normal hydrochloric acid that you are going to make. So make it accordingly. Here I am pouring the distilled water in the bigger measuring cylinder of 100 ml. So I need here 96.5. So after taking I will pour it in the beaker. So always observe the lower meniscus while measuring this type of liquid in the measuring cylinder, and then you pour it in the beaker. So preparation of one normal hydrochloric acid is done. The next step is for staining. So you need a watch glass. So you need a watch glass to stain the root tip that will be provided to you. So the next vital work that you have to do is you have to select the root. But in your examination hall, the teacher will provide you the root tip. Here I am taking one of the root tip from the bunch that I have already tied and made a bunch. So out of that bunch, I am selecting one good root, and I'm just plucking it out from the tied bunch with the help of a forceps, and then I'm cutting this tip of the root with the help of a scissors, and then I will stain it in the watch glass. So what is the staining procedure? That is, you have to take 9 drops of acetocarmine stain and 1 drop of 1 normal hydrochloric acid in the watch glass, and then you have to put it in the on the flame just to warm it, not do not boil it, just warm it. And after warming, you take that root on the slide and just gently place it on the slide in the middle and cover it with the help of a cover slip, and then you have to view under the microscope. That's it. So that is all you have to do for your activity of mitosis. So you can see I have put 1 drop of 1 normal hydrochloric acid in the watch glass, where I have already kept 9 drop of acetocarmine stain, and now I am placing the root tip into this mixture, and then I will warm the total thing over the flame, and then I will put it in the slide. So this is the way you have to do. So here I am keeping this root tip for a few moments and let it get stained, and then I am taking more than 1 root tip, so that I will prepare more than 1 slide, and I will see which one is giving a better result, and I will show you that one. So here you need some patience to do this activity. So let me warm the root tip. So warming process is on. Just warm for few seconds, even not for 1 minute, and then remove the root tip from the flame. So it's over. I'm removing the root tip from the flame, and now I will place it on the slide after it get cooled. Take a very clean slide and place the root tip in the middle of the slide, and after placing the root tip on the middle of the slide, see here I have taken 2 root tips, so I am removing another one and just placing 1 root tip on the middle of the slide, and then I will put the cover slip over it so that I can get a proper view of the cell division process that has taken place in the root tip of the onion. So I hope you remember which zone of the root undergo mitosis. Well, you can remember, and it's very important for your viva. Now it's time to put the cover slip over the root tip and view under the microscope. So gently place the cover slip so that there is no air bubble inside. Press it gently. Now there is one very important work that you have to do is, after you place the cover slip, you cover the slide by the help of a blotting paper or filter paper, and then gently tap by the back of the pencil or a pen on the slide. And even if you feel that the staining is not properly done or you need more stain, you can just put few drop of the acetocarmine stain just beside the cover slip, and will easily penetrate. So here I am placing and the blotting paper and gently tapping. Okay, so what happens when you gently tap the root that is placed on the slide? There are various cells. Okay, so the cells break, means the cell they spread across, and all the mitosis steps, whatever steps are there in every cell, means each of the cell might have been arrested in a particular stage of mitosis. Might be some cells are in telophase stage, some are in the metaphase cell. So this type of stages you can see. So I have already made the slide, and let me now view under the microscope. So you can see very well, so clear slide is it, and you can see the staining is done very properly. And now I am going to focus a particular cell stage. Let me clearly view, and then I can show you a particular stage. Well, here I can see some of the telophase Even I can see one. Well, let me take a close look. There are so many telophase stages are there. I will show you a still photo. Even metaphase can be viewed. Well, let me show you. So study the cell cycle very properly so that you can easily identify the stages. I hope you can see the metaphase here very well. You can see the metaphase and the telophase. So students, this time it is a 4-mark major experiment. So you are going to get 4 mark in this practical activity of slide preparation. So do well, and I hope you understood how to make the slide. Thanks for watching the video. Do subscribe my channel.